I will talk about transposon, which our DNA sequence that can move from one location in the genome to another. And this way they are known as a jumping gene. Look at how transposon works soon. Can working two different operation modes. In the first, they can jump from one location in the genome to another location, and in this process that do not leave any copy behind. This is known as non-repetitive modes of the transposition. In the second, they can make a copy of themselves, and the copy can jump from one location to another, is known as repetitive mode of transposition. So we can understand there are two modes of transposition. First is without duplication, and the second is with duplication. These things to be cut and paste and copy and paste mechanism. Transposable elements make up almost half of our genome, but are mostly silenced via different epigenetics mechanisms such as histone modification and DNA methylation. Transposon can be divided into three classes: DNA transposon, virus-like retrotransposon, poly-A retrotransposon. Let's talk about DNA transposon. First, which has particular region, and know the transposase code region which code for the transposase enzyme new part. Another feature is the terminal inverted repeats, which is the recognized by transposase. So let's see how they two work. So the transposase recognize the inverted repeats, the terminal, and they cleavage DNA, and they create its double-strand DNA break.
Its elements are involved in several diseases, like myocardial infarction of mental disorders, neurodegenerative disease. Microflora are the beneficial bacteria in the human body, which are beneficial bacteria at first and then turn into harmful bacteria later and cause disease. It is possible that this disease could be stomach cancer. Why did this bacteria turn from beneficial bacteria into harmful bacteria? It migrates to the genetic coding region of the bacterium, transforms it and produces a foreign protein, and causes